Let's look over exercise 29 from your lab book. This is all about functions of the heart. Remember, functions are physiology. So we'll look at how something works here. Whenever you get to chapters on neurons, muscle cells, or anything related to them, they often talk about these resting membrane potentials, depolarizations, and repolarizations. So we're on the heart, cardiac muscle cell. Let's do what we've got with this once again. Now remember, when you talk about resting membrane potentials, what they're telling you is this. When the cell is at rest, where are the potentials, meaning the charges, on the cell membrane? So when a neuron or a muscle cell is at rest, where are the charges at? Well, remember, there's only two charges, positive and negative. When neurons and muscle cells are at rest, they like to keep the positive charge outside, negative charge inside. So that's resting membrane potential. Positive out, negative in, right there. Now, where do these charges come from? They come from charged particles. Remember, that's what ions are. Makes sense that charged particles would be responsible for the charges. Now, the cells like to keep most of the sodium and calcium on the outside, most of the potassium on the inside. There's other things involved, but the balance of those three ions is primarily where that charge comes from. Now, let's say we want to depolarize a cell. Remember, depolarizing a cell means to swap the position of these charges. So that just means to bring the positive charge in. Now, when you bring the positive charge in, there's three big ways to do it. And this will generate an electric signal. Now, if you happen to be looking at a muscle as opposed to a neuron, this is when a muscle contracts. But remember, depolarizations and repolarizations don't cause muscle contraction and relaxation. They generate action potentials, which are electric signals. So let's look first at a depolarization. Remember what a depolarization is? To bring the positive charge in. Now, there's three big ways you might see cells doing this. They might open up sodium channels and let sodium come in. Look back when you see the neuromuscular junction with skeletal muscle, and that's what you see happening. Acetylcholine does that on the skeletal muscle cells. You might also see calcium coming in. Look at something like epinephrine and norepinephrine on cardiac muscle cells. Our cells could also close potassium channels and keep it in. So look what you're doing in either three cases. You're letting a positive in, or you're keeping more positive in. If you do any one or more of these three items, you're going to get up with more positive ion on the inside, and that's what makes the positive charge inside the cell at this point. That's depolarization. You swap the position of these charges by, instead of having more positive on the outside like you did here, now you got more positive on the inside. So that's how they swap position. Now again, this will generate an electric signal. If you happen to be in a muscle like cardiac muscle, this is when muscle contracts. Go down here to repolarization. Now repolarization is just the opposite of what we did. Now we're just going to move the positive right back out. What's the difference between that and resting membrane potential? Nothing, because that's what you're doing, is reestablishing the resting membrane potential. So if you want to repolarize a cell, do the opposite of what you did up here. Up here, you opened sodium or calcium channels and let it in, or closed your potassium and blocked those and kept it in. Here you do just the opposite. You could close those sodium channels and the cell will pump the sodium right back out. You could close those calcium channels and the cell will pump it right back out. Or those potassium channels that were closed, you can now open and let it out. Look what you did over here. You put more of these positive ions on the inside. That's what gave you the depolarization. Now you're putting more of those positive ions on the outside. That repolarizes right back where you started. Again, this is where you generate electric signals, but if you happen to be in muscle, like the heart, cardiac muscle, this is where the muscle contracts, and this is where the muscle relaxes, right here. Now let's look at this a little bit further and look specifically at cardiac muscle. So let's start with the sympathetic division. Remember, the sympathetic division of the nervous system is called that fight or flight division. Here's where you see sympathetic neurons releasing primarily epinephrine and norepinephrine. Well, what do those chemicals do when they get to your heart and get to cardiac cells? They specifically look for ligand-gated calcium channels and open them up. Well, if you open up the calcium channels, calcium's going to come in, right? Everything likes to move from high to low concentration, and plus this positive ion will be attracted to this negative ion material on the inside. So point being, these chemicals open calcium channels. Now look what you just did. You let some of that positive ion in. Now there's less positive out there than there was before. And that makes this outside charge less positive. You put some positive in here with this negative, and that's going to make it less negative. This is what's called decreasing the threshold, or hypopolarizing, right? And let's type that in right down here. Hypopolarizing a cell membrane. Let's type that in too, membrane. If you hypoed something, you made it less. So think about this again. By letting some positive calcium in, you took it away from that outside, making it less positive. Put positive in here with a negative, making it less negative. Now your charges are less different than what they were before. That means the cell has to move less positive ion in to swap them. That means it'll all happen quicker. And that's how these chemicals make your heart beat faster. It took those resting charges and made them less different. Now that they're less different, the cell can swap them quicker. You get a faster heartbeat. Now let's compare that to what the parasympathetic division does. Remember, this is called the rest and relaxation division. Now when this division here releases acetylcholine onto cardiac cells, it will look for ligand-gated potassium channels and open them up. Before we open calcium and let it in, now we're going to open potassium and let it out. Here we let a positive in, here we're letting a positive out with acetylcholine. Now if you move more positive ion out, this outside charge is going to get more positive. You take positive away from the inside, and that negative gets more negative. This is what's called hyperpolarizing. Remember, hypo made the charges less different. Hyper makes them greater in difference. So now that these charges have a greater difference than they did before, well, you got to move more positive ion in to swap them. Well, that's going to take more time, and the longer it takes to swap these charges, the slower your heart beats. So again, first thing we did with the sympathetic division and these chemicals were to make the charges less different. You can swap them faster, heart beats quicker. With acetylcholine, letting potassium out made your charges greater in difference. Takes longer to swap them, slows the heartbeat. This is how your body makes your heart beat faster or slower, letting a positive ion in or letting it out. So good information right there. You got to be really careful what you do with these three ions right here. You get an imbalance of these three, well, just look at one thing you might disturb. The functioning of neurons or muscles. Bad idea right there. So hope you enjoyed this information here. We will look at more further in the exercise.